Okay, welcome back everyone. This is lab presentation 7. And before working your way through this presentation, you should have already watched the video Life's Greatest Miracle, and you should have also labeled in your lab manual figures 52.1 and 52.2, and you should have become familiar with figures 52.3 and 52.4. And so let's go ahead then and start by looking at figure 52.1 on page 402. Take just a minute and orient yourself. And what you're seeing then is an ovary. So make sure you're finding that. And then notice leading away from the ovary, we're seeing a uterine tube or a fallopian tube or oviduct. And then we're seeing the fallopian tube leading into the uterus. Let's start then by watching what's happening when an ovary releases an egg cell with its surrounding supportive cells, remember. And so we're looking now at number 1 down in the lower left-hand portion of the diagram. And of course, you should have by now labeled this ovulation. And ovulation then is the release of that egg cell with its supporting cells. And notice then the figure is showing and telling you that fertilization typically occurs fairly high up in the fallopian tube. And when fertilization occurs then, the fertilized egg is now called a zygote. And so this is number 2 on your figure. And then notice if you continue to follow the arrows as this early embryonic structure continues to move down the fallopian tube to the uterus, we have mitosis going on. We have cell division going on. And take just a minute then, and first of all, find that zygote again. And then notice, and now we're looking at the series of cells that they're showing on the top part of the diagram. So notice now the zygote, the fertilized egg cell, undergoes mitosis and becomes a so-called 2-cell stage. And then as this 2-cell stage continues to move down the uterine tube, mitosis occurs again, and we go from a 2-cell stage to a 4-cell stage. The process continues then, and we go from a 4-cell stage to eventually an 8-cell stage, and so on. And notice then, we eventually have this structure called a morula. And the morula is still a solid ball of cells, but at this point, there are really too many cells to count them easily. You can't just glance at a morula, especially 3-dimensionally, and you can't go, oh, well, there are obviously 2 cells there, or 4 cells there, or 8 cells there. And so again, then, we use the term morula for this stage at which we have a fairly large number of cells, but we still have a solid ball of cells. And then finally, notice it's a late morula stage, which is now entering the uterus. And as you can see then, the morula next transfers, the morula then transforms into a hollow ball of cells called a blastocyst. And so number 3, then again, the late morula transforms into number 4, the blastocyst. And notice now within the blastocyst, which again is essentially a hollow ball of cells, but within this hollow ball of cells, we do have a clump of stem cells. And once you've located these, just jot down on your figure that this clump of stem cells within the blastocyst, this clump of stem cells is referred to as the inner cell mass. You can see then that the blastocyst implants into the endometrium, into the inner lining of the uterus. And much of what we've just talking about here should be familiar if you think back to watching that Life's Greatest Miracle video. Let's take a look then on page 403 at figure 52.2. And first of all then, I'd like you to find 3 of the 4 so-called extra embryonic membranes. And you might want to jot this term down. So again, we're looking at some extra embryonic, all one word, extra embryonic membranes. And as that name extra embryonic implies then, these are not part of the developing embryo itself. Again, these extra embryonic membranes, they're not part of the developing embryo. And let me give everyone just a minute to jot that down in your notes. So again, 4 extra embryonic membranes, and we're seeing 3 of them in this figure, in figure 52.2. And so go ahead and first of all then find the yolk sac, one of these extra embryonic membranes, and you can see then this is labeled number 1. Then find the chorion, labeled number 4. Take a minute now and just look at the extent of the chorion, and you can see that this membrane surrounds all of the other extra embryonic membranes. So you can see it's this large circular structure. And again then, notice it is surrounding all of the other structures that we're looking at in this figure. So surrounding the other extra embryonic membranes and also surrounding the developing embryo. And then finally, let's go ahead and find a 3rd extra embryonic membrane. So this is the amnion, number 5. Take a minute then and locate that structure. And note then that the membrane, the amnion, surrounds the amniotic cavity, which is labeled for you. Let's take a look now at the 3 so-called primary germ layers. And notice here again, the figure is not giving you that term, so you might just want to jot down there are 3 of these primary germ layers. And if instead of taking notes now for just a minute, you think back to the Life's Greatest Miracle video, you may recall that at one point we were looking at a single layer of cells, and then through a kind of a coming together and diving down and spreading out process, this single layer of cells transformed into 2 layers of cells. And then through this continued coming together and diving down and spreading out process, this 2-layer structure transformed into a layer of 3 cells. And again, these 3 layers then are the primary germ layers. And keep in mind, of course, again, with most or all of these structures, we're doing essentially anatomy here, and so you'll be hearing about these in lecture in terms of function. So those primary germ layers then, first of all, go ahead and find the ectoderm, and this is labeled for you. Then go ahead and find the endoderm, labeled number 3. And you may recall, again, thinking back to the video, this was the second layer to form. And I won't ask you that on a lab practical or on a quiz, but again, the endoderm, you may recall, with this diving down and spreading out, this was now the second layer to form. And then finally, the third layer, the third primary germ layer, is the mesoderm, number 2. So go ahead and find that structure. And then again, just thinking back to the video, you may recall this was the last of those 3 layers to form. This is the one that kind of squeezed in between the other 2. Let's go back to talking about the chorion now, and I'd like you to find the chorionic villi, and these are labeled number 6. And of course, by now, you're familiar with that term villi, implying finger-like structures, and you can see then that these villi, these chorionic villi, are these finger-like projections extending from a portion, from an area of the chorion. And here you might just want to jot down on your figure or in your notes that these chorionic villi are found in an area where the placenta will form. And we'll see this in the next couple of figures. But again, the chorionic villi are found in the area where the placenta will form and become part of the placenta. And then finally, just a couple of other structures that are labeled here. So take a minute and find the connecting stalk, which is number 7. And then also find the extraembryonic cavity, and this extraembryonic cavity is labeled for you. We'll stop here with this presentation then, and we'll pick it up next presentation beginning with figure 52.3 on page 404.